afternoon. How are you guys all doing? You all right? What have you guys been talking about for the last few minutes? I see someone's getting very agitated that we haven't posted the link yet. I can only apologize, but we'll send it out in a second. Someone is advertising another company. Lovely. Thank you for doing that in our time. That's very kind of you. What else we got going on? No, I think that's about it. That's about it. Anyway, how's everyone doing? Apart from wanting psychology as well, which I don't think we're doing any YouTube videos for at the moment. How likely do you think it is that teachers will predict year 13s any A stars in the summer? I know at least one teacher who has predicted one of their students an A star. So how likely do I think that is? If I know that there's at least one, I would say that it's a likelihood of 100% because I know at least one who will do that. But it depends. If you are the kind of student who's consistently got A stars throughout your years as a year 13 student, then very likely. If you've got like one, then probably pretty unlikely. What's up, Ollie? Hey, Sard. What's up? I don't know what's up. What have I been up to? Not very much. I watched a bit of Ashes to Ashes today. I went for a nice walk yesterday. It seems like certain people where I live are finding it very difficult to understand what 2 meters is. That has been something that I've noticed going on recently. What have you guys been up to? What have you done over the weekend? How's my day been? My day has been good, thank you. Very nice. I made, that was interesting. So you see all these people on like over the last few months been making loads of banana bread. I made some pierogi yesterday, which are like Polish dumplings. They're pretty good. Trying to hunt for an open McDonald's. I think you've got a long way that you're going to need to go. Yeah, so the one which was like the one that followed Life on Mars, Ash to Ashes, it's like police show. Nice. You're writing your classification and evolution notes, just to provide animal transport. That's weird, Emily, that you say that. I made a lesson today for like 3 weeks' time on classification and evolution. So that's odd. When do I think year 12 will be? What do you mean, when do I think year 12 will be? Is it lagging? Oh no, I hope not. What time is it here? The time is 2 minutes past 3. Yeah, do let me know if I'm lagging. It's kind of lagging. It's for you. Oh no. I assume if it's for one of you, it's going to be the same for all of you. Hopefully it sorts itself out in a minute. I don't know why it's lagging. My internet should be pretty good right now. Hang on. Yeah, I can't see why it wouldn't be very good. Hopefully it sorts itself out. Maybe it's just having a bit of a meh for a bit, and then we'll see what happens, eh? Anyway, so my plan today, in fact, let's just read a few of your messages whilst it's sort of temporarily fixing itself. What are you saying? Oh no, I'm sorry about slacking, guys. That sucks. How do you think year 12s will receive predicted grades because exams are cancelled? Well, I know lots of students are having mocks at the moment, so I don't know if you guys are having mocks. If you are, I guess I'll be from home, but I don't know. When do you think year 12 mocks will be? I don't know. Does anyone know if they have some of year 12 mocks? Yeah, so some students seem to be. So are none of you being told that you've got some mocks at any point? Or yeah, so that's what I've heard. I've heard some people getting them sent home and then they get like an hour to finish it from that deadline or something. So that's what I've heard. So it makes it very difficult to cheat, but I guess it'd probably be still possible to cheat. You got mocks as soon as you go back. 3 weeks. Only your biology teachers say back because biology teachers are the best. Mocks before they closed. Yeah, I think it's difficult, isn't it? Because you need your predicted grade so you can send off your UCAS things, don't you? So I don't know if maybe UCAS will be a bit later this year, but your school can give you predicted grades next year, I guess. So fees being the worst thing in the world. Your mock's supposed to be in April to May. Haven't heard anything from your bio teachers this whole time. You failed your mocks. Oh dear, oh dear, oh dear. Right. Yeah, hopefully this laggy thing, hopefully that fixes itself. But I can see that, yeah, on my one, it's been a bit slow. Maybe I've got too many tabs open. I mean, I doubt that I've got too many tabs open. But feasibly, I've got too many tabs open. I'm trying to see what it says my speed is. Oh God, it's never good when it's slow to start, is it? Oh no, apparently it's all right. My download speed is like 33 at the moment. So upload should hopefully it's going to fix itself and get better. Anyway, you're not supposed to be in April. Blah 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 blah. Very laggy. UCAT's still going on. Sound's gone off. It's frozen. Sound's gone. Back now. God, I can only apologise for this, guys. I'm sorry if this sucks. Supposedly my download's 40 and my upload's about 20. So I can't think of a reason why it's not working apart from, I don't know, maybe could be YouTube's end. It could be Zoom's end. Zoom was struggling. I don't know if you heard Zoom was struggling the other day. Oh well. Right, anyway. So I'm sorry, I'll start going through things, and hopefully because you're just going to see like little screens like this, the video doesn't matter so much. I guess my voice is a bit more important, and hopefully you'll be able to see some writing in a second, but we'll see what happens, eh? Hopefully it fixed itself. It could be Zoom. Oh well. Right. So some things to tell you guys very quickly. I have one final free account to give out. So I've been told that this week is the very final week that we are going to give out one free account. So I think 2 or 3 people have now got free accounts. I think I worked out the other day at least like 200 quid. So a free account up until September is about 200 quid's worth of free stuff. So if you are interested in this, all you need to do is follow us on Instagram, share a picture of your story, and basically tag Snap Revise. Okay. Some people managed to not do that very well. So they sent like a picture of me with like a wizard's hat on or something that said you're a wizard, Ollie. And whilst I thought that was really good, I'm pretty sure it wasn't actually on your Instagram or something. So I couldn't pick it. So I had to pick someone else, but I liked it. So yeah, if you want that, literally just follow us on Instagram, post a picture, and put it on your story, and that is it. Final thing as well, I've also got a coupon code for you as well. Some of you may have been able to work out what it's going to be. If you guess it right, then that'd be good. I think his computer has a virus or something. A pic of what? A picture of you enjoying yourself watching this YouTube video. So I don't know, maybe something about lagging could be quite funny. If you can think of a good lagging pun, that could feasibly be a good one. Your account needs to be public. If you send a photo of it, if you have a private account, we won't be able to see it, obviously. If you take a photo of it, like on your story, like screenshot it or something, that'll do. Yeah. Anyway, so here is the first question for you guys. So which of the statements, A to D, explains why diastole follows systole in the mammalian heart? So just in case any of you have ever heard your teacher say diastole and systole, it should be pronounced diastole and systole. Okay, so which of the statements explains why diastole follows systole? So cardiac muscle is myogenic. Cardiac muscle, oh God, I haven't even put the thing up yet. I'm such an idiot. Thank you, whoever just told me that I haven't got my screen up. You can just see my face. I can only apologize for that. There you go. So, sorry. Which of the statements, A to D, explains why diastole follows systole? So you guys can all see that. Yes, there you go. You can see that. And for those of you who are excited to see this, but I even drew big circles for you. You missed that. Oh, well. Gone. Is it because cardiac muscle is myogenic? Cardiac muscle takes a short time to repolarize after being stimulated. The aorta is capable of maintaining the pressure, or the SAN receives an impulse from the AVN. What do you guys reckon? I did like the wizard's art. It honestly made me laugh. I've got a picture of it on my phone still. I showed like my other friends. I think I put it on my Instagram actually. Okay, lots of you saying B. No one tempted to say A, C, or D? Benny and is. C. Okay. So in case you don't know them, which I'm sure all of you do, whoops, diastole is when the heart like relaxes. So why does the heart relax after it's contracted? Nothing to do with it being myogenic. That just means it beats by itself. Nothing to do with the SAM receives impulses from the AVN. That doesn't really have anything to do with it. And it's the other way around, if anything, isn't it? It's the AVN that receives an impulse from the SAM. Aorta is capable of maintaining pressure. No, that's nothing to do with it. Yeah, it has to be B. You guys are right. So the cardiac muscle takes a short time to repolarize after being stimulated. So some of you may not know this, you might, but essentially a neuron, after it's fired, needs to have a little bit of time sort of getting back to where it was. So you have something called the refractory period, and that is essentially what's going on. So it takes a little while before it all repolarizes for anything else to happen. Oh, yes. Finally, someone's asked me the 2 questions I normally get asked. In terms of specs, I always do the same specs, right? It's always going to be OCR, EdXL-A, EdXL-B, and AQA, the British ones. But things like CIE, things like Cambridge International, things like EdXL International, things like Educas, the Welsh one, I'm also covering those things too. I'm just not purposefully going through their specs with like a, I don't know, a fine toothbrush and stuff like that, right? So there's a good chance I'll be covering the things that you're doing, but I'm more focusing on the British ones. Also, other question, this will be an hour standard, but for this question, right, explain why diastole relaxation follows systole contraction. It's because after everything is contracted, the heart needs a few minutes just to sort of repolarize itself, I guess, to get it ready to contract again. So when it contracts, it is polarized, and in order to, or it's depolarized, in order to go back to what it was, it has to repolarize first. Okay, so essentially, it can't contract constantly for it to repolarize. Cool. Right, so which of the following, A to D, is the correct statement about tissue fluid? So tissue fluid carries carbon dioxide to the muscles. Oncotic pressure, for those of you that haven't come across that word, basically just change it to osmotic pressure. It's a bit different, but it's also quite similar. So oncotic pressure in the capillary causes tissue fluid formation. Hydrostatic pressure in the capillary causes tissue fluid formation. Or D, tissue fluid is reabsorbed into the capillary by active transport. What do you guys think? In Fuja, I will get there in a minute because there is one clear, obvious answer there. Yes, well done, guys. So it is C. So in terms of your capillaries, you know that they're very high pressure inside, well, there's a very high pressure in your artery, and it's quite high by the time it gets to the capillary. And essentially, the high hydrostatic pressure is enough to force fluid out. Now, oncotic pressure will move a little bit of fluid back in. However, the hydrostatic pressure to begin with is enough to overcome the oncotic pressure. So hydrostatic pressure is what causes tissue fluid formation. Essentially, for heartbeats, the pressure inside the blood vessels is going to increase, so much so that they can sort of leak out some fluid. Clearly, the best Harry Potter house is Hufflepuff as well, obviously. Okay, in the graph below, the top electrocardiogram, ECG trace, shows a normal heart activity, and the ECG trace below shows the abnormal heart activity. So some of you need to know this in more detail than others. It does come up for everyone, but some of you need it in a bit more detail. Slytherin is superior. Slytherin is like going based on the films. It's the one that all the horrible racist ones are in, right? We don't like the non-pure blood. Anyway, what is the heart condition represented by the bottom ECG trace? So fibrillation, tachycardia, ectopic heartbeat, or bradycardia. Is this OCR? Yes, OCR definitely. AQA, I'd argue, probably don't need to know it so much. EdXL, a bit more. AQA wouldn't expect you to know it, but AQA are a bit weird with this, where there's a page in their book where these are like ECGs are on it, but it doesn't necessarily say you have to know it off by heart. So I don't think they'd actually have it. Yeah, AQA wouldn't ask. AQA wouldn't have this question. So tachycardia, what does that mean? What's tachycardia? Let's get rid of some of the easier ones. Fast heart rate, yeah. So tachy is fast. So it's definitely not, well, to be fair, it is fast. But what you normally would expect with a heartbeat is you have certain sort of regions. So you have a P wave, you have this thing in the middle, which is a QRS complex, then you have a T wave after, right? This isn't showing that normal heartbeat, so it's definitely something that isn't tachycardia. What's bradycardia? Yeah, you guys are right as well. So ectopic is where you get like, if you've ever had that feeling in your chest where you've like skipped a beat, your heart just hasn't beat, or it's done one additional one and it just feels weird. That's an ectopic heartbeat. This is not showing you an ectopic heartbeat. There's not one additional one. Bradycardia is slow, so we're left with fibrillation. So it's got to be fibrillation. Essentially, fibrillation kind of means quivering, and you've got this weird, like, kind of strange sort of wave. You don't have any visible P waves here. You basically just have this QRS complex going again and again and again. This one's really serious. If you had an ECG and your heart looked like this, that'd be a problem. You definitely need to go to the doctor pretty quickly. Yeah, tachy just means fast. So yeah, this isn't good. If this ever happens, get some help. Like, I don't know how you know this is happening, but get some help. Cool. A student studied the structure of a blood vessel and found that an outer layer of collagen. They found an outer layer of collagen, a thick middle layer of smooth muscle and elastic tissue, an innermost layer of endothelial cells. Which of the options A to D identifies the type of blood vessel that the student studied? Ectopic heartbeat, Massimi, is where you have like a bonus beat, or you have like, you miss a beat. Bradycardia is where it's slow. It's just a lot slower. So most of you are going for A here. One of you is going for D. So, okay, let's cross some things out. So outer layer of collagen. So I know that one of these doesn't have collagen, so it can't be a capillary. They don't have collagen, right? They just have endothelium on the outside. A thick middle layer with smooth muscle. So which one has a thick, smooth muscle? Basically, one of them does and one of them doesn't. Yeah, it's got to be an artery. So arteries have very, very, very thick, smooth muscle. Does anyone know why that's the case? Why does it have such thick, smooth muscle and such thick elastic? What bit maintains blood pressure, the elastic tissue or the muscle? See, everyone says pressure, but that's not actually right. So it's got loads of elastic to maintain pressure, right? And if you feel your pulse, that's what you're feeling. So hang on. If you guys get your two fingers and you get your arm, and the side that has your thumb on, if you put your fingers there and you push, you should be able to feel your arteries getting slightly wider. So essentially, the elastic is there so they can stretch and recoil. Right? Essentially, that is there to maintain pressure. The stretching and recoiling will maintain pressure. The muscle, the smooth muscle, on the other hand, isn't there for anything to do with pressure. So the muscle is there to control the flow of blood. So, for instance, I have pretty much in the last half an hour had my lunch. For those of you who are interested, I had some super noodles and they were delicious with some sriracha sauce. Essentially, all of my energy at the moment, well, not all of it, a significant portion of my blood is going to my digestive system so that I can digest food and then absorb the nutrients. So all of the muscles with arteries going towards my digestive system are going to be vasodilated. The muscle will be relaxed. All of the not all of them, lots of the arteries going towards my legs and to my arms, they are probably not going to be being used right now because I'm just sat down. So the muscles, the blood vessels, the arteries going to, say, my legs are going to be vasoconstricted more than they'd usually be because my legs don't need as much blood because I'm not using them. Yeah, soup and noodles. I didn't have soup and noodles. I had super noodles, which are like the cheap noodles, like the ramen noodles, but cheap. Cool. So venule is a small vein. Yeah, venule is a small vein. Arterial is a small artery. Interestingly, and this is something that everyone always forgets, arterioles have more smooth muscle than an artery. Okay, and the reason for that is because they control the flow of blood a little bit more. Vasoconstricted means that the muscle is contracted. So vaso meaning blood vessel, constriction, a lot smaller. Vasoconstriction equals contracted muscles. So it's going to have a smaller diameter. Vasodilation is where they relax and you're going to have a wider diameter. Okay, so if you want blood to go to your brain, then you've got to hope that all your blood vessels going into your brain are going to be vasodilated. It's so weird to have toast in the afternoon. It is weird to have toast in the afternoon. Can anyone no longer see Ollie? The camera has just gone. I've disappeared, have I? That's bizarre. I promise you I'm still here, don't you worry. Oh, I'm back now. I honestly don't know what's going on. It's got to be Zoom or YouTube, because my internet's really good. Like really good, like bizarrely good, like never this good. Oh, you'll see my hair is very long at the moment and I'm getting frustrated. I can't do much about it. Right, anyway. So the pressure changes in one mammalian cardiac cycle are shown in the graph below. Which of the following time periods, A to D, shows ventricular systole? Okay, so let's have a look. Which of these showing me ventricular systole would you guys guess? Just sort of off top of your head. So far, these have all been multiple choice, haven't they? Right, it's getting harder soon, don't you worry. So thick wall is control blood flow, exactly. Does muscle control? Yeah, muscle controls vasodilation or vasoconstriction. So B, most of you guys are saying B. So what does B say? So, right. Obviously, we need to work out when ventricular systole is. So this line, it's a bit stupid that they haven't done it as a dashed line. They should have done it as a dashed line, really. It's a dumb question, if you ask me. But obviously, the time when ventricular pressure is going to be highest is going to be ventricular systole. So it starts in here about 0.2, and the pressure stops being high about here. So it's going down from that moment onwards. So this bit here is probably going to be diastole. Okay, so it looks like it's going to about 0.3 in my mind. So I would say 0.2 to 0.3. There you go. Yeah, I'd say it's B. Yeah, systole means contraction. Okay, A, yeah, no, definitely not that. C, 0.4, no. 0.6, no. Cool, lovely. Right, first hard question then. So describe and explain how substances that are dissolved in the blood plasma, such as oxygen or glucose, enter the tissue fluid from the capillaries. In your answer, you should use appropriate technical terms spelled correctly. So how does glucose say, or how does oxygen, no, let's go for glucose. How does glucose enter the tissue fluid? How does this work? No, you don't have to have a public account. No, I would never see me. I'd never expect you to put your security at jeopardy. Just make sure that you've taken a photo and put it on your, I don't know, what's it called, your story, and then screenshot that and then send it to Snap Provise. It's got to be a way to do it, like DM us. See, I'm down with kids, I know the words. So by diffusion, what does that mean? So what's moving by diffusion? What's high to low concentration gradient? Good. So it leaves a capillary down the hydrostatic pressure gradient. Okay, what leaves a capillary down the hydrostatic pressure gradient? So hydrostatic pressure pushes glucose. No, hydrostatic pressure pushes something else out of the capillaries. When am I going to pick? I'll probably wait until Wednesday. So I get the rest of my colleagues to have a look. We all have a good goggle, and we go, oh yeah, that one's funny. Or in my case, someone sent me like a not Harry Potter, like a wizard one. And we're like, yeah, that's a good one. Hang on. So what are we saying? Capillary beds surrounding cells allowed nutrients. Yeah. So lots of you guys are telling me that oxygen is forced out, right? What is actually forced out of my capillary? Water. Water is pushed out, isn't it? So let's draw that diagram, right? So here is my capillary, right? There's some water which is moving out. How does glucose get out then? So water's going to move out. Why does the glucose come out with it? Good, yeah. So I think that's an important point to make. Glucose is dissolved in water. Or you could say oxygen is. Okay. Now, how does water get out then? So we said that the glucose is dissolved in the water. How does the glucose get out? So hydrostatic pressure is higher than oncotic pressure. So tissue fluid is forced out of the arterial end of the capillary. Exchange occurs on the tissue. So that is a good start. Okay, so John, what are the other forces then? So hydrostatic pressure is one force which is trying to push out the water. What's the force that you said was going in? What's the force pushing things back in? So good. There is oncotic pressure too. Maybe I should write that out in full. So hydrostatic pressure. And then this one over here is oncotic pressure. Now, I said earlier, for those of you that weren't here, that if you do AQA, for instance, they don't actually use this word oncotic pressure. They just say like osmosis, but I'm going to write osmotic pressure or osmotic potential or something like that. Yeah. No, AQA doesn't actually use oncotic pressure. It just means water moving by osmosis, basically. It's basically saying that there's a net inward movement kind of thing. Anyway, so vasodilators, when we refer, you're talking about ACE inhibitors, United one. You could do for that, yes. Anyway, so glucose is dissolved in. What's the other pressure? So oncotic and hydrostatic pressure. What are the other pressures? There's two other ones. ACE inhibitors. ACE inhibitor, ACE inhibitor. AQA does make life a bit easy. Aortic pressure, not really. So just one thing to think about, right? The other way. Hydrostatic pressure in the blood vessel is pushing it out. There is also a very small hydrostatic pressure trying to push it back in, but the net hydrostatic pressure is going outwards. So in questions like this, I would probably say the net hydrostatic pressure is going outwards, and I'd say the net oncotic pressure is going inwards, because there is a little oncotic pressure going this way as well. But on the whole, the oncotic pressure going inwards is slightly more. So I would just start to try and say maybe net. Although I don't think it quite makes sense for oncotic pressure. It makes more sense for hydrostatic. But I think I'm making this more confusing than it needs to be. So glucose is dissolved in water. High hydrostatic pressure at the arterial end. And then we want to say something like forces fluid out or forces water out. So forces water out. You can also get a mark if you mention the oncotic pressure. So you might want to say something like the oncotic pressure at arterial end is lower than the hydrostatic pressure. Okay, so I'm looking for one more mark here then. What could one more mark be? So I've got 3. Can I go over what hydrostatic pressure is? Yes. Hydrostatic pressure is essentially a means of looking at the amount, well, not really actually. Basically, it's kind of like gas pressure, but with a liquid instead. So the more water there is, the more hydrostatic pressure there is, the more sort of, oh, that's not a great way of saying it either. Essentially, when the ventricle, so say the heart's up here, right? As soon as the ventricle beats and blood is sent out of my aorta, essentially that blood is pushed very, very, very hard. So the ventricle is pushing it really, really, really hard, which forms the pressure, right? So it's kind of like if you have a water pistol and you like pump it up and then you spray it. The hydrostatic pressure inside that water pistol is very high, which causes it to get like spurted outwards. So the heart basically builds up a massive pressure when it contracts. And remember, it contracts upwards. So all of this blood is sort of forced out at a very high pressure. That's basically what hydrostatic pressure is. So over here at the top end, the arterial end, it's much higher than it is at the venial end. Right, I need one more mark here. So one more mark. So why can it actually leave the artery? Sorry, the capillary. Why can the water actually leave? So oncotic pressure is basically just osmosis, right? There's always an oncotic pressure going inwards because there's loads of water outside in the tissue. Because it's super thin. Good guess, Catherine. Not quite. Molecules are small. The molecules are small and good. Capillaries are very leaky, right? So white blood cells can move out of a capillary. So we want to say glucose is dissolved in water. There's a high hydrostatic pressure at the arterial end. The oncotic pressure at the arterial end is lower than the hydrostatic pressure. Then we want to say the capillary, not the capillary, You have a capillary. The capillary is naturally leaky, or you could say permeable. Allowing small molecules out, such as water. Okay, so for those who are a bit confused, then hang on, I might try and draw this again. All right, so there is your answer, pretty much as nicely as you could put it, if I do say so myself. Right, so essentially what you've got then in terms of the tissue fluid is you've got like your little capillary that looks like this. In fact, let's change the color of it so it stands out a bit better. Bear with me. Right, here is my capillary, right? Now, this is called the arterial end. This is called the venule end because that's what joins to a vein. This is what joins to an artery. In the tissue fluid, so in this fluid around, I've got all these cells, right? So the tissue fluid is basically the liquid that is covering the cells. So all of your cells are bathed in this sort of liquid. So I could, oh God, this diagram is going to get insane. Just you bear with me for 1 second. Ooh. Here we go. Right, so here is this sort of fluidy stuff. So this blue stuff is meant to be water, right? So there's a lot of water surrounding my cells. The cells are the little purple things. It's on both sides because why not? Can you tell that I didn't do art at GCSE? Can you tell that I decided to give that one a miss? You guys are probably too kind to say that. Anyway, so like the moment it becomes a capillary, yeah, exactly. So the arterial end is the very moment that the arterial turns into a capillary. It's just given the name of the two ends just to make it a bit easier to think about. Right, so inside my blood vessel, let's do a little bit of water, but not very much. Your blood vessel has a bit of water in it, to be fair. So here's a bit of water in it because it's plasma, remember. But on the whole, it's mostly going to be red blood cells. Right, that's the main thing which is inside of your blood vessels, as I'm sure all of you know. There you go. There you go. Right, so at the arterial end, the thing that you need to be aware of is that the pressure inside that artery is very, very high. So the pressure is very, very, very high because it's just, that's weird, it went black. It's very high because basically the heart has just contracted, systole has just happened, ventricular systole, and that's caused the high pressure. So that's going to be a net force pushing the stuff in the vessel out. Now, on top of that, there is another pressure which is pushing something back in. So oncotic pressure is basically there is a greater water potential outside in the tissue fluid than there is inside. So there's a little bit of water that's moving in. So most of the water is going out because of the high hydrostatic pressure, but a little bit of that water is trying to move back in because there is a greater water potential outside than there is inside. By the time you get down to the bottom, the hydrostatic pressure is actually very low. So the hydrostatic pressure is quite low at this point because it's all been used up earlier. But this oncotic pressure has stayed the same as what it was. So there's a little bit of water coming out, a little bit, not a lot, but a little bit, because there's still some hydrostatic pressure. However, the oncotic pressure is greater than the hydrostatic pressure, so water moves back in. So essentially, to summarize, when there's a high hydrostatic pressure, water goes out. Oh, that's a good way of saying it. When hydrostatic pressure is greater than oncotic pressure, liquid's going to move out, and that happens at the arterial end. When oncotic pressure is higher than hydrostatic pressure, water will move back in, and that happens at the venule end. So if that's what you want to remember, that's a good way of remembering it. Right, if you do AQA and you don't have oncotic pressure, just say like water potential or osmotic pressure or something like that. Okay. And again, it's not very hard to remember as well, because you know that it leaves at the top and it comes back in at the bottom. So if it leaves at the top, that's where the pressure's got to be highest going out. And if it comes back in at the bottom, that's where the pressure going in is going to be higher. Right. Describe and explain how the wall of an artery is adapted both to withstand and maintain a high hydrostatic pressure. So for 5 marks, how is the wall of an artery adapted to withstand and maintain a high hydrostatic pressure? If you do AQA, can you get marks saying oncotic? Yes. If you do AQA and you use the word oncotic, that is the correct word to use, so you'll get the mark. Okay, what does the thick muscle do? What does the elastic tissue do? So, right, okay. Let's say very thick elastic tissue. What does the very thick elastic tissue layer do? Very thick elastic tissue. Withhold the pressure, recoil and stretch. That's what I want to see. Good. What's it actually do, though? It does what to the pressure? One of the words that they've said earlier. Does it withstand or maintain? Not cope with. I want to... Not to actually withstand. The elastic tissue is to maintain. So very elastic. Very thick elastic tissue to maintain pressure. And then the reason for that is because, in fact, let's make that a bit more, a bit further down. Go away. As it can stretch and recoil. So essentially it stretches when the beat happens, which will help to, I don't know, increase the pressure it can sort of have or something. In fact, it's going to decrease the pressure by stretching, and then it recoils, which is going to increase it up a little bit again. So essentially, the elastic tissue is there so that the pressure inside is roughly the same. It doesn't change too much. Now, it obviously does change quite a bit, but it would be more if you didn't have this elastic tissue. Okay, so that's what it does. Someone said collagen. Where do you find collagen? Good. So collagen, which some specs mention, others don't. The thick outer wall. What does that do? Maintain or withstand? How does it get out of the sides of the capillaries? They happen to be leaky. There are little gaps between the endothelial cells. Good. Collagen, thick outer layer, thick outer wall, withstands pressure. So it stops it from bursting. Oops. For some reason, I've written out 2 words, but I'm sure you guys can cope. Withstand pressure. Very good. What else? Yep, so elastin I've done, collagen I've done. Smooth muscle. Smooth muscle is a weird one. Do you need to know about collagen for AQA? You actually don't need to know that collagen makes up the thick outer wall. If you do AQA, just say thick outer wall. Can I repeat the question's answer? I haven't done all the answer yet. Muscle layer is thick, so the volume of blood passing can be controlled. So yeah, weirdly, they actually accept this. So thick muscle. Now, I wouldn't accept this if it were me, because it's more about controlling blood flow. It just confuses people. But they have given thick muscle can constrict. What will that do to the pressure if it constricts? AQA biology is really good. Collagen, I think, is mentioned at Excel, but it might be at Excel A and not B, or B or not A. Good. So thick muscle can constrict to increase pressure. So therefore, if it's going to increase it, is it trying to withstand or maintain it? Good. Maintaining pressure. Okay, what else? Any other things that we could say? Any other things that you know of? Lumen. Now, I don't know if they actually give anything about the lumen. I don't think they'll let you have that. Oh, apparently they do give... Oh, no, they don't give that as mark. The size of a lumen, I guess you're saying that it's narrow, which is going to help to maintain a high pressure. I guess that's what you're saying, right? Now, weirdly, they don't actually give that as a mark. Something they do give, which some of you might not know, I'll give you this one, is the endothelium is slightly folded. In fact, oh no, yeah, let's go for that. So endothelium is folded, basically allowing it to stretch a little bit. So it's got little folds in it, and this is going to help to maintain pressure. So there you go. There's one last mark. If you said about the lumen being thin, I think they might have accepted it, but they didn't on this question that I've got here. So I think you might be okay. Yeah, Google it. Google the folded endothelium of a artery, and you'll see it. No, so the endothelium, right? It's the very middle bit. So that's your endothelium. So this is the lumen. This is the endothelium. This bit here is going to be your smooth muscle and your elastic tissue. And then this is your outer sort of collagen layer. Yeah, the lumen's a whole. Okay. Large animals, such as mammals, need efficient transport systems. Figure 3.1 shows a section through the mammalian heart. Name the parts labeled X, Y, and Z. Was the second to last word in the third point. Withstands. Sorry, this was meant to be me saying withstands, but I wrote it as like 2 words because I'm an idiot. Okay, X is which atrium? Oh, you said it. It is the right atrium, yes. Well done. Y? Ooh, pulmonary artery or aorta. Look at the side this on. Yes, the aorta. So weirdly, they're crossing over in this diagram, which doesn't help. You normally don't see it like that. But because this blood vessel here connects up to this side of the heart, which I know is the left, it's combined into the left ventricle. So it's got to be the aorta, which means that Z is what? Yeah, it's a pulmonary artery. Now, you could be more pedantic here and say what lung it's going to go to, which would get you, it could feasibly get you a mark. So it might be worth saying it. Which lung is it going to go to? Yeah. So it's your left pulmonary artery. Right now, some of you might not have ever considered this, but obviously each lung is going to get a different blood vessel going towards it, isn't it? So this one's going to go to my left lung. This one's going to go to my right lung. So you didn't need to say left, but if you wanted to, I don't know, could be another thing that you could say. Do you need to know this for AQA? Yes, you need to know this for AQA, yes. Structure of the heart, 100%. Ooh, good question. So explain how pressure changes in the heart bring about the closure of the atrioventricular, otherwise known as the bicuspid valve. So if you ever get stuck on tri or bicuspid, something my teacher once said to me was you learn to ride in fact, let's just draw. If you want to draw the heart, this is how I've always drawn the heart. So if your blood vessel coming down there, valve there, blood vessel coming down there, valve there, valve there, blood vessel going in. There was one blood vessel coming out. There's one blood vessel going out. Another blood vessel going in there. There you go. There's a nice heart. So this one, this is my right side, this is my left. You learn to ride a tricycle before you learn to ride a bicycle. So tricuspid, bicuspid, easy. Otherwise, atrioventricular. So explain how pressure changes in the heart bring about the closure of the atrioventricular valve. How is it going to close? So higher ventricular pressure than atrial pressure. Very good. Yeah, good. Well done. 2 marks though. So what else can we say? So pressure in the ventricles. Higher than atria. That is 1 mark. Which happens during ventricular diastole, systole, which happens in ventricular systole. So that could be another mark. You can get some marks for some other things. So it happens after ventricular systole. Nothing to do with the SAN. When pressure in the atrium ventricle, no, not quite. So what are some things that prevents it from folding back on itself? Does anyone know what the things are that prevent your valves from inverting? Lob is during, systole. Dub is during, caudae tendineae is what they're called. So the little heartstrings that I'm sure your teacher referred to them as when you did your lesson, that sort of look like that. You could mention them. Tendons prevent inversion or something. You could say that. Interestingly, you could also get a mark if you said cusp-shaped valves. Right, I'm sure lots of you don't know what I mean by that. Essentially, if this is a blood vessel and this is a valve, right, blood can go through this way because it can literally just push the two valves apart in the middle. So you'll get some tendons down here, some tendons there. However, if blood tried to go this way, it would build up here and it would build up here and it would literally force it closed. It forces it closed here, right? So the cusp-shaped valves is what I've just shown you. Basically, blood can't flow backwards because it can't, basically it fills up the two valves of blood and they literally physically can't open. So it can only open in one direction. Yeah, so diastole is when you get the dub sound, which is the second sound. Nothing happens during atrial systole. Ventricular systole is when you get the lub sound. So your heart's like lub dub, lub dub, lub dub. No, nothing to do with thickness. So yeah, basically during diastole, which one's going to close? So your semilunars are going to close during diastole. So this is semilunars closing. This is AV closing. So when you hear your heart beating, most people think that's the actual beat that you can hear. It's not. It's the sound of the valves closing. Okay. Right. Next one. How many more do I have? I've got loads more. Blooming heck. Right. I might not have time to do all of these, but I'll do as many as I can. Blood clots can form if the lining of an artery becomes damaged. So not everyone needs to know this. AQA don't, for instance. OCR do a little bit. In fact, OCR don't actually. Edexcel A do, Edexcel B do. So for each of the statements below, put a cross in the box that corresponds to the correct statement about the blood clotting process. So a platelet does what? How come the valves are different? Tricuspid has 3 bits to it, bicuspid has 2 bits to it. I don't know why it's the case. It's just some weird evolutionary quirk, I guess. Ooh. So C or D, or D, or C, or A. So what do platelets do? What are platelets? Everyone should know this because you all did GCSE biology, hopefully. Or at least science. Yes, so it is C. So your platelets, they make thromboplastin. So they make thromboplastin. Thromboplastin is the one, in fact, is that my next question? Yes, kind of. Thromboplastin converts something called prothrombin in the cascade to thrombin, and then thrombin does something else, right? So this is what thromboplastin does. Thrombin is what though? So is thrombin an enzyme that changes fibrinogen into fibrin, an enzyme that changes fibrin into fibrinogen, an insoluble protein that forms a mesh to trap red blood cells, or is it a protein that sticks to collagen in damaged walls of arteries? What do you reckon for this one? What is thrombin? Olivia's got an answer. United 1 has got an answer. Molly's got an answer. Yes, it is A. You guys are correct. So thrombin converts fibrinogen, which is soluble, into fibrin, which is insoluble, so that you can form your mesh, basically where it traps red blood cells and platelets and all that stuff. Okay, so a stroke can be caused by cardiovascular disease, CVD, affecting arteries leading to the brain. Callum's family has a history of strokes. Sorry if any of you are called Callum. Explain why a blood clot in an artery leading to the brain could cause a stroke. So clots that we've just done are not in AQA, those of you asking. It is in Edexcel A and B, and it's slightly in OCR, but not very much. This one is relevant to everyone. What's a mesh? Like lots of fibers all over the place. Have you ever looked at a knitted jumper? That's a mesh, not a mess. Yeah, sorry. So why could a blood clot lead to a stroke? So this question, I think, is relevant to everyone. Yeah, mesh spelled M-E-S-H. Good. So it doesn't stop it entirely, but let's say reduced oxygen flow and glucose. Why is there less oxygen and glucose? Good, because there's reduced blood flow. What's the problem with that? Why do you need oxygen and glucose? Oh, Olivia said it. So less, don't say none. Oh, we get out of the habit of saying none, by the way. Say less whenever you have a question like this, because there's almost always going to be a little bit, but you want to say less. So less aerobic respiration can carry on. Why is aerobic respiration so important? Why does the brain need to do aerobic respiration? Don't say respire, say aerobic. If you're saying that there's less oxygen, say that it's less aerobic. Don't be lazy. What kind of energy? In the form of... Yeah, it's going to prevent the buildup of lactic acid. That's actually a mark. Good. So less ATP can be synthesized. Right, you could say less lactic acid is, more lactic acid is synthesized. Too bad, get you a mark if you said that lactic acid inhibits enzymes or something, which is fine. Okay, ooh, right. Come on, I really want to finish this. I've got like 8 minutes or so to finish. Read through the following passage on the cardiac cycle and write on the dotted lines the most appropriate words. So the cardiac cycle consists of 3 stages: atrial systole, ventricular systole, and diastole. During atrial systole, I'm enjoying me spelling here, guys. The atria, both atria. What I'm saying, atria, not atrium. The atria contract and the ventricles are relaxed. Lovely. The something valves are open. Good. The atrioventricular valves are open. The atrioventricular valves are open. During ventricular systole, the semilunar. So during ventricular systole, the semilunar open as oxygenated blood is forced out of the heart through the aorta to the body and through the pulmonary artery. Very good. To the lungs. Easy question. Describe 2 differences between the structure of a capillary and the structure of a vein. How are the veins and capillaries different? We've managed to do that last question in about 30 seconds. Come on, guys, let's do this. I believe. Good. Which one has valves? Capillary has no endothelium. Unfortunately, capillaries are made up of just endothelium. Good. So, right, let's say that. Capillaries are 1 cell thick. Well done for answering this properly. So capillaries are 1 cell thick, whereas veins consist of multiple cells. Good. Capillaries don't have valves. Well done. Everyone always forgets that veins have valves. Capillaries don't have valves, whereas veins do. What else? Veins have more elastic tissue than capillaries. Good. Capillaries don't have any. You could say that capillaries don't have any collagen, whereas veins do. You could say that capillaries don't have any smooth muscle, whereas veins do. You could say that the lumen size is different. So you could say that capillaries have smaller lumens than veins. You could say capillaries are more porous, or you could say they're more permeable, or they have a leakier a leakier surface than veins, or something like that. Do capillaries carry any deoxygenated blood? Yeah, at the venu end they will. So I wouldn't say that. Right, 2 more questions. Let's do it. Many animals, such as mammals, have we done this? No, we haven't. Many animals, such as mammals, have a heart and circulation. This helps them to meet their requirements by overcoming the limitations of diffusion. Describe the structure of the mammalian heart. Good. 4 chambers. What else? Good. So you need to tell me a little bit more about the atria and ventricles. Where are the atria? You need to tell me where the valves are as well. Where are the atrium and ventricles? Anyone? Good. Okay. Atria on top. Ventricles on the bottom. Okay. What else are you saying? Myocardium. Ooh. If you no, I don't know if you've said very much. Okay. So thickness of wall. So what side's thicker? I think someone did say it. Ooh, septum separates the two sides. Left from right. Good. Left side's muscle is thicker than the right side. How good would this question be? Can you imagine how happy those kids were when they saw this question? Tricuspid on the right. Yes. So you could say atrioventricular, AV valves separate atria and ventricles. Semilunar in the blood vessels or in the arteries. You could say that tricuspid is on the right, bicuspid is on the left. Right, what else could you have said then? So some other marks. You get a mark for saying where the SAN is. So if you said that the sinoatrial node is found in the right atrium, you'd get a mark. If you said that the AVN, the atrioventricular node, is found towards the middle of the atria, you'd get a mark. If you mention coronary arteries, you'll get a mark. If you say where all the veins are, you'll get a mark. So vena cava is on the right-hand side. Pulmonary vein is going to be on the left-hand side. Position of arteries will get you a mark. Tendons will get you a mark. If you say it's made up of cardiac muscle, you'll get a mark. And that's about it. So I've got 6 there. Yeah, anything else is all good. You won't get it for myogenic because it's a structure and not function. Can you draw a diagram for this? Oh, yeah, I reckon if you drew a really good diagram here, you'd get quite a lot of marks. So I would be tempted to probably do that. Right, as I said, draw a heart like this. So blood vessel 1, blood vessel 2, tendon. Sorry, that valve, tendon, blood vessel 1, blood vessel 2, 3, 4, done. Right, final question. Cardiovascular disease is responsible for many deaths. One cause of CVD is atherosclerosis. Some of you need to do this, some of you don't. Describe how atherosclerosis forms. Myogenic means it beats by itself. Is a mark scheme to these on Snap Provides? No, there's no mark scheme on Snap Provides. I've just gone through all the answers. Thrombosis is to do with this, yes. What happens first? What's the start of atherosclerosis? Oh, so those who don't do this, AQA don't do this, for instance. Not in much detail anyway. I think OCR do it a little bit, but not a lot. EdXLA do it a lot. EdXLB do it a lot. Good. So damage to the endothelium. If you can tell me what causes the damage, that would be good. So damage to endothelium, normally due... Ugh, that was weird. That was so weird. What just happened? Oh, that's weird. Anyway, due to high blood pressure or smoking or something. So you could say that nicotine and smoke causes it. This causes a inflammatory response. What happens with inflammation? OCR do it a little bit, not a lot of detail. Like very, very little detail. Like as in you wouldn't need to know all this. Good. Build up of white blood cells. And release what's it? Cholesterol, which I've apparently forgotten how to spell. I think so. So white blood cells build up and release cholesterol. This is what causes an atheroma. Atheroma means basically fat. So it causes an atheroma to form. After that, you get the formation or calcium salts are released. So calcium salts are released, forming a plaque. Okay, this is going to cause a reduction in lumen diameter. Let's say volume. Reduction in lumen volume, which is going to equal higher blood pressure, which is going to be positive feedback if you wanted to say that. And then we want to say something along the lines of loss of elasticity and hardening. There you go. Something like that sounds good. So no marks of fatty tissue. If you said an atheroma, that is fatty tissue. So you could feasibly have said either. Right, what are you guys saying? Do we need to talk about atheroma rupture to trigger thrombosis? No, you wouldn't need to say that it could. That is something that's a bit different. So you're talking, about it basically bursting. You're talking about an aneurysm, which is the word for that. So it becomes narrower. Yeah, no marks of fatty tissue, probably not. That could lead to a clot by the plaque. Yeah, so exactly. Right. Can anyone tell me what the link is between a valve and snap revise? Does anyone know? How's a valve similar to snap revise? What do valves do? What does snap revise do? Opens doors. Oh, I like it. Know what I was thinking. Too smooth. No, that's good though. That'd be like the endothelium. They stop blood from going to the wrong chamber. Let's try and make it. Oh, they prevent backflow of knowledge. Oh, that is exactly what snap revise does. So this tube here, this is your journey. I like to refer to it as. This is your journey, right? And you are somewhere over here. Here you are, having a nice little time. Don't know. Maybe you're like quite happy over here. Look at that happy person. And then here is your academic success. Now, there are many things that are going to get in your way as you move through here. Maybe there could be the equivalent of, I don't know, some other bully in your way or something. That's a terrible example. Maybe there's like a pressure against you. Oh, there we go. Maybe there's a pressure against you. And some bloke has decided to call you silly or stupid, and you got very cross with them, right? So they're in the way. On top of that, maybe you have other people in your life who are getting in the way, or maybe the pressure is put on us by society to look and conform to certain conditions. Maybe they're pushing you in a certain direction away from academic success, teaching you that instead of that, maybe I want to have a really peachy bum or something, right? And that's going to get in your way. However, snap revise, the only reason I said that is because I heard one of my friends' brother is now doing fitness. No, don't worry. But anyway, what snap revise is a cusp-shaped valve, right? So here are some snap revise valves. Here they are. Okay, what they do is they allow you to get to the next stage. So you're going to have to do some work. Don't get me wrong. We're not going to just tell you everything. But you're going to do a little bit of work and you'll get to this next stage, right? So you're no longer here. You're now here. Look, you've grown a bit as well because as an individual, snap revise will help you to grow. You're now here. If there's a backwards pressure on you, well, the furthest back you can now go is here. Almost like a lifeline on Who Wants to Be a Millionaire. I'm a bit like Chris Tarrant, actually, in that sense. But you've pushed back a little bit, but not very far. Let's say that you do a bit more snap revise. Ooh, you've got through to the next stage. You've got that £250,000 question, correct? Right, here you are, right? Essentially, what's going to happen if there's a bit of backwards pressure? Oh my God, I think I'm going to die. No, I'm not. Right, if there's a bit of pressure, you can't go back. Snap revise's tendons is going to stop you from inverting, and you're going to get closer and closer and closer to academic success. So this is what I'm offering you. I'm offering you a system that's going to prevent you from going backwards and going to keep pushing you forwards, but it's going to be independent. So for those of you who don't know what snap revise is, let's pick, what should we do? Let's look at OCI level biology. If you were to sign up, not only would you have a source which has pretty much a video for, in fact, no, let's go even further than that. Apparently I've done this quiz too often, like I'm not allowed to do that quiz anymore. Let's look at the genetic code, right? Not only do we have a system to find out what you do know and what you don't know, which will help to be that barrier to prevent this backflow. Not only do we have that, which then tells you, for instance, after you've done it, where you went wrong and which videos to then watch. We then have videos which talk over the things you need to know. It will tell you down here when you've completed it, and you might, for instance, not get it, and someone like me will be able to answer. But you have the videos, and then following the videos, you have a little quiz. So let's see how you've now done. Hopefully the valves of Snap Provide will kick in and prevent you from going backwards any further, right? Let's say you've done that, and you're now like, oh God, I wish I had an exam question. I blooming wish I had an exam question. Well, there is some bloke who'll go over a exam question with you, and he'll talk you through how to do it. And then he'll show you what the answer is to that exam question. There it is. And then he'll do another 2 of them. So you'll have 3 exam questions, right? Let's say after that you thought, wow, I've seen him do it, but can I do it? Well, the valves of Snap Revise will buy it again. Here are a load of exam questions for you to have a go at doing. And, oh, what's the answer? I wonder what the code is that goes with that. Bam, there it is. Here is what you need to know. Look, here are the answers all written down nicely for you so that you don't have to go anywhere to find them, but they're right here. Then you can say that you've got it, right? Not only that, because that would be enough by itself in my mind, but it goes one step further. There's a revision guide that you could then have a look at. Here is a lovely revision guide about proteins that you could stick on your wall or something, keep forever. And lovely. Like, after all that, how could you possibly go wrong? Like, what else could there be? Well, my friends, I tell you, there's a little bit more. So maybe that wouldn't be enough. And maybe you'd be like, oh, I kind of get it, but I still don't get it very well. Well, at this stage, this is when web classes at the very top end of Snap Provide kick in. So if you wanted to, you could come and see me teach considerably more. So as opposed to just twice a week, I'm doing about 10 a week or so-ish. So I've got antibiotics practicals, which I'm tomorrow. I've got on Wednesday, basically some math skills. I've got some osmosis things going on here. I don't have anything on Friday. And then I have a drop-in tutor after that, where on Wednesday, so not tomorrow, on Wednesday, I've got a drop-in session where people can just turn up and ask to go through things. And I've only put 2 of them on my calendar at the moment because I forgot to actually prepare myself. But I do at least 2 of them every week, as well as a book club. So going back to my little analogy, which I'm sure all of you are very bored of by now, essentially, Snap Provide is here to help you along your journey by preventing you from going backwards, essentially. But how much does it cost, Simone? That is a good question. How much does it cost? I would say it costs a fraction of the price of going along your academic journey. No, it costs, there are different packages. So the cheapest one's about £10 a week, and that'll get you like the videos. The most expensive one's about £50, not a week, £10 a month. The most expensive one's about £50 a month, and that's one which has all of like my online lessons as well. So to be fair, the most expensive one is quite expensive, but for what you get, it's really, really not. So I used to charge about 40 quid per hour of my time when I used to tutor, and this is like a fraction of that. So for those of you that are interested, here is a £10 off code, cardio10. Unlucky if you guys didn't manage to work that out. Fabulous, I think, is about how you've written it. £10 a week, no, £10 a month. I misspoke, sorry. So the cheapest is £10 a month. The most expensive is £50 a month. But that's the most you're ever going to get. And you'll get a lot of like one-on-one. You'll get a lot of like sessions with me basically as your teacher. What else were we saying? Can it be free? You can get a free account. If you go to our website, you can get like a taster bit and you get a few videos for free just to see if you like it. I'm brainwashing you. Sorry. Does Snap Provide reply to comments? On YouTube, sometimes. On Snap Provide's website, every single comment, yes. So much lag. I'm sorry about the lag, guys. That is really annoying. Annoyingly, it hasn't come up. Well, I haven't noticed it so much, but obviously for me, for you guys, it has. Ollie, could you hold out your hands beside you like you were holding a box? Yes, medium size next to your head, please, for like 10 seconds. Gone. Like here, I'm really concerned that this is going to be horrible, but gone. Like, hang on. Hopefully that's enough. I'm so terrified of what you're going to do. Right, anyway, what else are you doing? What's in the middle package? So the middle package, I think someone will send you a link. Hang on. If you scroll up to where it says snapprovides.co.uk/plans, you should have something there which tells you what you get. So the package are these. I'll just show you. Save you having to go to the bother. Boosh. Right, so you have videos with the basic package, exam walkthrough videos, and mini revision guides. Oh, that's it. So the pro package is the one where you get all the quizzes, which mark themselves. You get the predicted exam. Actually, you know, I showed you all those exam questions. You get all of those. And for the other one, you get, I don't know, something else. Oh, sorry. And then for this one, you get the web classes, drop-ins, and tutor support to chat whenever you want. Okay, so that's what you get. Oh, yeah, final thing as well. If you want to post the Instagram thing, last chance to get a free account for the package, the ultimate package with the teaching, feel free to do something on Instagram and I will find it and I will see what I think. Okay? Any other questions? I'm glad this is helping with your biology though, guys. To my left, please. Oh, right, okay. So my left, so you mean this side, yeah? That's my left. I'm terrified. Once more. I'll do that side too. There you go. So anyway, is it together middle or best package? Well, obviously, I think the best one is the one which has my teaching videos, but I guess it depends how much you get from my teaching videos. If you don't enjoy my teaching, probably don't do that one, I suggest. If you feel like you're getting quite a lot from it, do that. Yeah, you can cancel as well after, if you've had 1 month and you thought like, oh God, this guy sucks, feel free to cancel. Well, so a vaccine is essentially a dead or attenuated version of a virus. Why is it so hard to make a vaccine? Because it's not hard to make one, it's hard to test if it works. And to test if it works, you need to test it on loads of people and make sure that they're not doing any better than the placebo. That's why it's hard. Out of curiosity, where did you go to uni? I went to the University of Southampton for my degree, and then for my postgraduate teacher stuff, I went to UCL. Excellent. I'm looking forward to seeing the story at some point in future. Right. Anyway, I'm going to probably head off now because it's already like 1/4 or nearly 1/4, and I probably should have ended this like 12 minutes ago. So have a lovely rest of your day, guys, and I'll catch you on Wednesday. See you later, Miss Amy. No worries, Maria. No worries, Sarah. No worries, Dr. Cocktail. Interesting name. Right. See you later, guys.